Chapter 4 will explore the particulars of bacterial growth. In order to understand bacterial growth, we have to look at how a population of bacteria increase in numbers. Bacteria divide by an asexual reproduction technique called binary fission. In binary fission, one bacteria divides, producing two daughter cells. One bacterial cell divides, and the result is two daughter cells. When each of the two daughter cells divide, the population will now have 4 bacterial cells. When each of the 4 bacterial cells divide, the population will have 8 bacterial cells, which will in turn divide to make a population of 16 bacterial cells. 1, 2, 4, 8, 16. These doubling in number of a population is called exponential growth. Bacteria divide by exponential growth, quickly increasing their numbers by doubling the population every said length of time. The length of time that it takes for a population of bacteria to double in numbers under specific conditions is called generation time. In this particular example, the generation time of this population is 30 minutes. So the generation time is 30 minutes per generation or per division. The first time the population divides, during the first 30 minutes, the population increases from 1 cell to 2 cells. During the second generation, which takes place during the second 30-minute interval, the population doubles in numbers, increasing from 2 cells to 4 cells. During the third generation time, which occurs at the next 30 minutes, the population will increase from 4 cells to 8 cells. During the fourth generation, which occurs at the next 30-minute interval, the population doubles in numbers again, going from 8 cells to 16 cells. So again, generation time is the length of time that it takes for a population of bacteria to double in numbers or to go through a generation. Generation time is the time it takes for a population of bacteria to double in numbers. A generation is the actual doubling in numbers that the population experiences. Generation time varies among the species and is influenced by environmental conditions such as temperature, availability of nutrients, or availability of moisture. For example, the generation time for some Mycobacterium species at optimum conditions is 24 hours per generation. Compare that to the generation time of E. coli, which at optimum conditions is 20 minutes per generation. Which of the two organisms grow faster? Generation time changes as environmental conditions change. If we want to slow down E. coli's growth, we could change the temperature at which we're growing it. Or, if we want to increase its generation time, we may want to increase the temperature and nutrients. Exponential growth results in rapid population growth. Bacteria divide by exponential growth. Their populations doubling in numbers every generation time. For example, 10 cells of a foodborne pathogen in potato salad at a picnic can become 40,000 cells in 4 hours, provided the generation time is 20 minutes per generation. This is due to exponential growth. Arithmetic growth would result in 22 cells, one cell added to the population every 20 minutes. Given the formula for generation time and two of its parameters, we could easily calculate the third. For example, if we know that the generation time for a specific bacterial population is 0.33 hours per generation, which translates into approximately 20 minutes per generation, and the incubation time is 4 hours, we could ask how many times does the population double in numbers during that 4-hour period? How many generations or doublings occur in the population during the incubation time? We know the generation time, we know the incubation time. Using the formula for generation time, we can solve for number of generations to know how many times the population doubled in numbers. If we solved for number of generations, we get number of generations equal time of incubation over generation time. Now we can plug in the two parameters that we do know in order to solve for the third parameter. We know the population incubated for 40 hours. We know the generation time of the population is 0.33 hours per generation. Hours cancel. 4 divided by 0.33 generation gives us 12.1 generations. So in 4 hours, the population of bacteria doubled in numbers 12 times. Let's look at another example. Let's say we know the number of generations that transpire during a specific amount of time, 6 hours. In 6 hours, the population doubled in numbers 10 times. What is the generation time? Well, we know that generation time is the time of incubation over the number of generations. All we have to do is plug in the two parameters we know, time of incubation over number of generations, to calculate that the generation time for the population is 0.6 hours per generation, or a little over 30 minutes per generation. Another formula that can be used to calculate different parameters of bacterial population growth is NT equals N0 times 2 to the N, where NT is the number of cells in a population after a set amount of time. N0 was the original number of cells in the population. N is equal to the number of divisions or the number of generations. So using this formula, for example, we could calculate the final number of cells in a population of 10 bacteria that go through 12 generations. NO is the original number of cells in the population, which is 10 in this particular example. N is the number of generations, which is 12 in this particular example. Remember, we had 3 divisions per hour for 4 hours. NT equals 10 times 2 over 12. 12 being the number of generations. Solving the equation gives us 40,960 cells of the pathogen in 4 hours. Let's look at another example. If there are 20 Staphylococcus aureus cells present in a tuna sandwich, and the sandwich is left in a warm car for 3 hours, how many cells will there be at the end of the 3 hours if the organism divides every 30 minutes? This is what we have. 20 is the initial number of cells in the population. 3 hours is the time of incubation. 30 minutes per division is the generation time. 30 minutes per generation can be converted to hours, which will give us 0.5 hours per generation. order to calculate the final number of cells after the 3-hour incubation, we need to use this formula. We have the original number of cells, the initial number of cells. However, we do not have the number of generations. We could calculate the number of generations by using the generation time formula. Remember, generation time equals time of incubation over number of generations, which now we're writing as n. If we solve for n, we have that n equals time of incubation over generation time. We have time of incubation as 3 hours. We have generation time as 30 minutes per generation, or 0.5 hours per generation. 3 hours, incubation time, 0.5 hours per generation, generation time. As we plug in the numbers, notice that hours cancel, leaving us with generations as the only unit. 3 divided by 0.5 will give us 6 generations. N equals 6 generations. Now we have all the needed parameters. We have N, we have original number of cells. We can now calculate final number of cells, or NT. NT equals 20 cells times 2 to the 6. 20 times 64 cells will give us 1,280 cells at the end of the 3-hour incubation. You should now be able to answer the following questions. If a population has a generation time of 0.5 hours per generation, how long will it take the culture to reach 10 generations? Which organism grows faster at 37 degrees centigrade? One with a generation time of 0.5 hours per generation, or one with a generation time of 0.35 hours per generation? What can be done to slow down the growth of a population of microorganisms?